Hey, what's going on? This is Eric Olson for Sciencentric, and today we're talking about making babies. No, not the classic way of making babies. This is the modern way of making babies, and it's caused quite an uproar in the scientific community with good reason. Specifically, we're talking about the news out of China that a scientist there has created a CRISPR baby. So if you don't know what CRISPR is, that's a way to directly edit a cell's DNA structure and fundamentally change it. So let's head over to the NIH website. I think they have a really great description of how CRISPR works before we go any further, and then we can talk about the ethical issues with fundamentally changing people's DNA. Okay, so the NIH website describes CRISPR in this way. CRISPR-Cas9 was adapted from a naturally occurring genome editing system in bacteria. The bacteria capture snippets of DNA from invading viruses and use them to create DNA segments known as CRISPR arrays. The CRISPR array allows the bacteria to remember the viruses or closely related ones. If the virus attacks again, the bacteria produce RNA segments from the CRISPR arrays to target the virus's DNA. The bacteria then use Cas9 or a similar enzyme to cut the DNA apart, which disables the virus. So this is basically a defense mechanism that bacteria use to prevent viruses from invading. It's similar to a little bit to how our immune system works and how our cells work in remembering when viruses attack. This is why vaccination works, for example. So the CRISPR arrays are basically little snippets of the viral DNA or RNA that have actually been incorporated into the bacteria's genome. So the NIH website goes on to say the CRISPR-Cas9 system works similarly in the lab. Researchers create a small piece of RNA with a short guide sequence that attaches to a specific target sequence of DNA in a genome. So basically, this artificially created RNA, instead of attaching to a virus, it's attaching to whatever piece of DNA the scientist wants it to be attached to. The RNA also binds to the Cas9 enzyme. As in bacteria, the modified RNA is used to recognize the DNA sequence, and the Cas9 enzyme cuts the DNA at the targeted location. Although Cas9 is the enzyme that is used most often, other enzymes can also be used. Once the DNA is cut, researchers use the cell's own DNA repair machinery to add or delete pieces of genetic material, or to make changes to the DNA by replacing an existing segment with a customized DNA sequence. So this is like a really easy way for scientists to make changes to individual cells, to the DNA in individual cells. They basically are making a cut. They can put in whatever kind of genes or pieces of genes that they want, and then the cell's own machinery goes in and sort of knits the DNA back together. So that's essentially what's happening. It can also be used to make deletions so that you can cut out a piece of a gene, a problematic piece of a gene, and make the gene more functional and work better than it did with that problem. So the difference between this and other forms of genetic engineering that have existed in the past is that this is a very specific way to introduce changes to an organism's genome. And in the case of what happened in China, now we're talking about we're fundamentally changing the DNA of a human, an individual, and that DNA can then be passed on if that individual decides to have offspring. So this just opens a huge can of worms, a Pandora's box, if you will, because once these changes are introduced, they can't be taken back. So let's hear from the scientist who did this, and he has a very thick Chinese accent, so it might be a little bit difficult to understand, but I think it's worth hearing from him directly. So one thing to note is that it's my understanding that this scientist, his name is He Jiankui. Pardon my Chinese pronunciation if that's way off. My understanding is that this is the way that he told the world about what he had done, that he had fundamentally changed the genetic structure of these babies, and he announced it on YouTube. So it's highly unorthodox. Usually when you publish new research, you're going to publish it through a peer-reviewed academic journal, not announce it to the world through a series of YouTube videos. So I think it's interesting to hear from him, and you can definitely hear the excitement in his voice about this discovery.
Two beautiful little Chinese girls named Lulu and Lana came crying into the world as healthy as any other babies a few weeks ago. The girls are home now with their mom Grace and dad Mark. Grace started her pregnancy by regular IVF.
Okay, so I just want to stop the video there. So he says that the babies came into the world as healthy as any other babies, and the truth is that we don't actually know that. We don't know that those babies are 100% healthy. They may look healthy, maybe born looking healthy, but their genes have been altered, and at this point, only time will tell how healthy they are. So I think he's perhaps overly optimistic here.
Right after we sent her husband's sperm into her egg, we also sent in a little bit of protein and instruction for a gene surgery. When Lulu and Lana was just a single cell, this surgery removed the doorway through which HIV entered to infect people. A few days later, before returning Lulu and Lana to Grace's womb, we checked how the gene surgery went by whole genome sequencing. The result indicated that the surgery worked safely as intended. Grace's pregnancy was normal, which we monitored closely by ultrasound and blood tests. After birth, we again deep sequencing Lulu and Lana's whole genome. This verified the gene surgery worked safely. No gene was changed except the one to prevent HIV infection. The girls are safe, healthy as any other babies. When Mark saw her daughter for the first time, he said that he never thought he could be a father.
Okay, so the reference to HIV there is that the gene that he changed is a gene called CCR5, which is involved in how HIV, the virus that causes AIDS, enters into the immune cells. So the idea here is that the father had HIV, and by changing the genetic structure of the babies, the embryos, that would prevent the entrance of HIV, I guess, into their cells. Although I'm not 100% sure how that works. I'm actually a little bit surprised that they can't separate out sperm and egg from any kind of HIV viruses during the process of in vitro fertilization. That's actually kind of surprising to me. So I'm not exactly sure what the point was here, other than to try to create immunity for these babies from HIV. But it's not 100% clear to me that they would have been infected to begin with, because they were selected by IVF. So it seems like that could have been screened out some other way. All right, so let's jump over to an article by Gina Colada and Pam Bellick at the New York Times. And they have a nice article that's about why are scientists so upset about the first CRISPR babies. And actually, they have a nice synopsis, too. A Chinese scientist recently claimed he had produced the world's first gene-edited baby, setting off a global firestorm. If true, the scientist has not yet published data that would confirm it. His actions would be a sensational breach of international scientific conventions. Although gene editing holds promise to potentially correct dangerous disease-causing mutations and treat some medical conditions, there are many safety and ethical concerns about editing human embryos. Here are the answers to some of the numerous questions swirling around this development. The scientist He Jiankui said he used CRISPR, a gene editing technique, to alter a gene in human embryos, and then implanted the embryos in the womb of a woman who gave birth to twins in November. That is illegal in many countries, including the United States. China has halted Dr. He's research and is investigating whether he broke any laws there. Among the concerns are whether the couples involved in Dr. He's research were adequately informed about the embryo editing and the potential risks involved. Dr. He says he has submitted his research to a scientific journal, but nothing has been published yet, and he has announced the births of the twins before his research could be peer-reviewed by fellow scientists. So then they get into a little bit, as I was alluding to, is, why did he edit this CCR5 gene? They write that the gene is called CCR5. It creates a protein that makes it possible for HIV, the virus that causes AIDS, to infect people's cells. Dr. He said that with the help of HIV/AIDS advocacy organization in China, he recruited couples in which the man had HIV and the woman did not. He used the CRISPR-Cas9 editing technique to try to disable the CCR5 gene in their embryos with a goal, he said, of creating babies who would be resistant to HIV infection. So again, I don't really completely understand what he was trying to do there, because this was not a natural insemination. It was done by IVF. So it seems like you could screen out any, HIV particles that would be floating around in this man's semen before creating a IVF baby. So why are scientists up in arms? Changing the genes in an embryo means changing genes in every cell. If the method succeeds, the baby will have alterations that will be inherited by all of the child's progeny. And that, scientists agree, is a serious undertaking that must be done with great deliberation, and only to treat a serious disease for which there are no other options, if it is to be done at all. Instead, Dr. He went ahead and disabled a perfectly normal gene, CCR5, while people who are born with both copies of CCR5 disabled are resistant to HIV. They are more susceptible to West Nile virus and Japanese encephalitis, and there are simpler and safer ways to prevent HIV infection. So I think that this is probably the most fundamental problem with tinkering around with genes in embryos, not individual cells of an individual, but actually changing the genetic structure of future generations. There's a phenomena in biology called Pleiotropy. This is basically the idea that a single gene can affect multiple traits. So if I go in and I try to change a gene and make somebody resistant to HIV or resistant to some other kind of disease, they may have some unintended effect because I'm affecting some other trait, as they point out in this article. If I try to make someone smarter, it might make them shorter, or more prone to breast cancer. I mean, we just don't know. And anybody that's worked in developmental biology knows that these genes affecting one gene can have multiple effects. I spent a little bit of time in a fruit fly lab, and we would change changes in one gene could have a ripple-on effect through the development of the whole fruit fly. With basically birth defects, mutations that would give the fly extra legs or extra joints or something like that. So when you start tinkering around and you don't know exactly all the everything that's going to happen, it's a really bad idea. And you want to start with animals that you can destroy if something ends up wrong with them. There's so many ethical issues with people, because obviously we can't, experiment on people and then destroy them if we get a bad result. It's like once that mutation is introduced into the germline, we don't know what the effects are, and we don't know how that will persist in the population. We don't know how it could affect other traits, and to top it all off, we can't put it back. And consider this: what if these babies that grow up to be adults want to have children? Can we tell them that they can't have children because they're genetically inferior in some way because of these mutations that have been introduced? I mean, that just sounds totally draconian and totally authoritarian and just a terrible idea. So, I think the science community is outraged about this because we really have to proceed with caution when we're talking about editing what's known as the germline, the cells that eventually become egg and sperm, because we just don't know what's going to happen down the road, and we have to proceed with great caution. There may be some instances where we can use genetic engineering like this to eliminate diseases from the population, but we have to be really, really careful that we're not then introducing mutations that have effects that we're just not aware of. All right, so let's go back to the New York Times article. More worrying, CRISPR often inadvertently alters genes other than the one being targeted, and there are also some circumstances called mosaicism where some cells contain the edited gene and others do not. Dr. Hay, I don't know how to pronounce it, claimed in a video that CRISPR did not affect other genes in the twins and that the babies were born normally and healthy, but there is no way to know that is true. In fact, some of the data Dr. He presented at a conference in Hong Kong after he announced the birth of his twins is concerning. Several scientists said, for one thing, it indicates that he was able to disable both copies of the CCR5 gene in only one of the twins, whom Dr. Hay identified as Nana. In the other twin, Lulu, only one copy of CCR5 was disabled, providing limited, if any, protection against HIV. But Dr. He implanted the embryo anyway. He said he informed the parents and they wanted both embryos implanted. Some scientists said the data Dr. He presented also suggested several potential issues resulting from the editing process. Most importantly, said Dr. Kiran Musunuru, a geneticist at the University of Pennsylvania who reviewed the data, there's clear evidence of mosaicism in the edited embryos of both twins. I was so furious, Dr. Musunuru said, this would have been disturbing anyway. Gene-edited babies. It made it 100 times worse knowing that he had totally mosaic embryos. It's as if you took the embryos and dipped them in acid and said, you know what, I'm just going to go ahead with the implantation anyways. It's not that much different. So in this case, CRISPR was applied to embryos, not individual cells. And so some of the cells may have been more or less affected by the CRISPR-Cas9 system, meaning that some of the cells did get, DNA inserted or DNA deleted in this case from them, and some did not. And, that's not, I'm not exactly clear what the problem is with mosaicism. I think from a scientific perspective, it's just sort of not a clean experiment because you don't know which cells of the body are going to end up having been affected by the CRISPR system, CRISPR-Cas9 system, and which ones aren't. So, then you don't know, are these babies actually, resistant to HIV or not? Other than that, I'm not exactly clear what the issues with mosaicism are. If you're a geneticist and you have information about that, I'd love to hear from you. Either hit me up on Twitter at sciencentric, or if you're watching this on YouTube, in the comments. I'd like to know. I'm not 100% clear on that. All right, so let's go back to the article. While it's unclear if the babies themselves ended up with a mosaic patrick of cells, Dr. Musunuru said the data shows that Lulu's placenta was mosaic, which is not a good sign. Finally, it is not known if his study subjects knew what they were agreeing to. The consent they signed was for an AIDS vaccine development project, and it did not mention all the risks of disabling CCR5. It said that if CRISPR altered other genes, the project team is not responsible for the risk. Okay. What are the potential implications? Many scientists are concerned Dr. Her's experiment could have a chilling effect on support for legitimate and valuable gene editing research. So should such epic scientific misadventures proceed, a technology with enormous promise for prevention and treatment of disease will be overshadowed by justifiable public outrage, fear, and disgust, said Dr. Francis Collin, director of the National Institutes of Health. So I think, truth be told, that this CRISPR-Cas9 gene editing thing to most people probably sounds like science fiction and isn't even on their radar. So, I mean, I know that there's definitely pushback against GMO foods. So, genetically modified foods and, GM people, that's probably just beyond what most people can even comprehend. I mean, there are definitely some issues. And, it's not, because this sort of thing is actually pretty easy to do from a scientific perspective. I think it's going to be really, really hard to regulate this in any way. But let's go back to the article because this is addressed there as well. So in the United States, Congress has barred the Food and Drug Administration from even considering clinical trials involving human embryo editing. The National Institutes of Health is prohibited from funding such research. The National Academies of Science, Engineering, and Medicine issued a report in 2017 concluding that editing the genes of human embryos should only be acceptable in the narrowest of circumstances. It would have to be used to correct a serious genetic disorder that causes disease or disability. There would have to be no other alternatives. There would have to be good evidence that the benefits would outweigh the risks, and there would have to be a plan in place to follow the gene-edited children. In a follow-up video, the researcher in China, Dr. He, actually released 5 different videos, and in one of the videos he addresses that and kind of lays out his own ethical guidelines for gene editing. What's odd to me, though, is that it doesn't seem like he actually followed his own advice, and these children, again, as far as I can tell, were not at any risk of contracting HIV. If they had been naturally conceived, that probably would have been a problem. But I think with IVF, there's probably a way to filter out HIV before the egg and sperm are joined in a test tube, and then implanted. So I just I think that is a good sort of thing to have, a good ethical guideline, which is to only use this to correct genetic problems, not to use it to modify people to make them better or in some way by making them taller or smarter or better looking or free from male pattern baldness or whatever. I mean, that's a slippery slope, and as I mentioned before, I mean, pleiotropy and sort of downstream effects are a serious concern. What are the ethical concerns? Some worry that this is the first step toward using gene editing to create people with extreme intelligence, beauty, or athletic ability. But that, for now, is not possible. Such traits are thought to be affected by possibly hundreds of genes acting in concert and affected in turn by the environment. The biggest ethical concerns for now are with the rogue scientists enticing couples who do not realize the risks to babies that might result from experiments. And when those children grow up, the altered genes will be passed on to their children and to their children's children for generations to come. What we still do not know, until Dr. He publishes the results of his work in a peer-reviewed medical journal, we will not know the detailed results of the embryo editing or even whether the twins were actually born. Dr. He has not submitted his data, nor has he identified the children or parents other than to provide first names for the twin girls, Lulu and Nana. These may be pseudonyms. We won't know for many years if CRISPR affected the genes other than CCR5, nor can we gauge the health of the babies now or in the future. And of course, we do not know if other scientists will be emboldened to try their own experiments editing the genes of human embryos. So I think that this was bound to happen. We knew that, this sort of experimentation was happening in monkeys, in other primates, which are, our closest biological relatives. It was only a matter of time before somebody sort of took the first step and took the plunge, so to speak, and just dove in and actually tried to do this. So the problem I think here is that science is a global endeavor. If we can't do it in one country, some scientists in another country will take it on. So I don't know how this is going to be prevented or regulated. This is going to be a global effort with governments cooperating across the globe, and regulate, a worldwide ban or regulation on this, I think is going to be difficult. And, it's more difficult than something like, regulating the production of nuclear material, because this is much more accessible and easy to pull off. So I don't know. I don't know what the solution is. Let's take a look at Nature News. The Nature News team put out an editorial suggesting how to move forward from this. Let's look at that for some ideas. All right, so the article says, people like to say that science is self-correcting. Events in China last week pose a serious challenge to that reassuring platitude. How do researchers respond to the failure of medical ethics, collective responsibility, and professional standards that saw an immature experimental technique used to help produce human babies? It has not yet been independently confirmed that the Chinese genome editing researcher altered the DNA of embryos using a gene editing technique and then implanted them in a woman, as he claims. Such a step would be significant and controversial because it would make a permanent change to the germline that could be passed on to future generations. This distinguishes germline editing from the use of gene editing tools as therapies that correct genetic alterations in somatic cells in blood and other tissues. Somatic cells are just the regular cells of your body that don't give rise to egg and sperm. Verification of He's claims could be difficult given that privacy concerns rightly protect the identity of the parents and their 1-month-old twin girls. But many scientists in the field agree on two things: the relative simplicity and widespread availability of the gene editing tool CRISPR-Cas9 mean that what He claims to have done is eminently possible, and whether or not he is the first person to have genetically edited a baby, he will not be the last. I think that's 100% true. So although testing the accuracy of his claim is a priority, so too is ensuring that any future efforts to genetically edit the germline of human babies proceed in a much more regulated and responsible way. The scientific community still has the opportunity to take the lead on this. Public and political reaction to last week's news has been calmer than many might have expected, and it should do so urgently. So I'll just jump in and say that I think, again, that this is just off people's radar. I think if you ask most people what CRISPR-Cas9 was, they would just have no concept of what it was. So the fact that there's genetically modified babies out there now, it's just not on people's radar. It needs to be. There should be a wider political reaction. I think people don't understand the ramifications. They don't understand that this is like the genie that can't be put back in the bottle. And so if it catches on and more people are doing this, it's going to be, we just don't know how that's going to turn out. We just don't have enough information. So again, we need to proceed with caution. Okay, back to the Nature News editorial. Some argue that the circumstances in which germline gene editing would be beneficial, such as reverse disease-causing mutations that could not be addressed in any other way, are likely to be extremely rare. Nevertheless, given that research and medicine move fast, a clear regulatory system needs to be devised and put in place in case a credible proposal arises. Such a regulatory system should draw on those that already exist to guide the use of gene editing tools for research into human development, and more broadly, govern medical testing of innovative therapies. But it should not start with the assumption that future germline editing is a foregone conclusion. That is a question for society, not scientists, and one that demands the input of different stakeholders from across the world. Researchers and physicians must ask permission rather than beg for forgiveness. So I think that's a nice idea, but the truth is that this researcher in China, I mean, he was first, and he will always go, assuming that this is true, that he actually accomplished this, but that, he will always be first. He will have the infamy, he will be famous, or he will have infamy regarding this, and his name will always be attached to it. And that is the nature of science. You need to be the one that does it first, and it's what drives research forward in a good way, but it's also can drive it forward in a bad way, and because people are just doing things sort of thoughtlessly and carelessly. Not most scientists, but there are ones out there who are sort of unscrupulous. So again, that's a nice idea, but something stronger, I guess, may be required here. And maybe it's a matter of monitoring materials that would be used for something like this. In the same way in the US they monitor materials that could be used for explosives, just so we know at any time who's doing this kind of work. All right, so let's go back to the Nature News editorial. So a solid regulatory system set up by the research community can then be the basis for laws and regulations that individual nations might decide to introduce. Debate was key to framing the law that regulates a mitochondrial replacement therapy in the United Kingdom, a procedure that also affects unborn babies and means they carry DNA from 3 people. Laws are not always the best way to govern emerging medical procedures, but they do offer the deterrence of effective punishment for those who don't follow the rules, unlike self-regulation or guidelines. So how can the gene editing community set up a better system? A starting point would be a global registry or national registry set up by the funders or governments to record clinical research that involves gene editing in human embryos. This would require the objective steps and limitations of projects to be spelled out from an early stage. The record should also detail the steps taken for ethical approval and oversight of the research. The 2016 guidelines from the International Society for Stem Cell Research are a good model to follow for regulation of research that involves human embryos and gametes, including research into germline editing. So again, that sounds like a great idea, and I think for ethical researchers that would be a good first step. But I think there will always be those researchers, those scientists, or just unscrupulous people that want to do this outside of any kind of regulatory system. So how do you identify them, and how do you make sure that they're that this isn't happening somewhere else? I mean, it does take some technical skill, but I think a regulatory system, a voluntary system, is not going to catch everything or everything that's going on. So there may need to be laws that are also act as some kind of deterrent. So the Nature News article goes on to say, such registries could also provide a mechanism to flag research projects that do not meet high ethical and technical standards, and a route to apply pressure on individuals and their institutions to improve, and they could provide a framework, if the time comes, to define a path to the clinic. They would help to explain the risk and potential benefits to people such as prospective parents, so they can make more informed choices. Her's claim to have communicated his intentions and actions to the scientific community do not stand up to serious scrutiny. The community, from individual researchers to institutions, can and must do more to encourage more meaningful, transparent engagement and discussion on specific projects. In return, scientists who are trusted to carry out research have the responsibility to welcome and embrace scrutiny. I mean, yeah, absolutely. He did this work, didn't tell anybody he was doing it, kind of came up with a PR campaign by some accounts. He hired an American PR firm to help him, create sort of a campaign and videos, and then, announced to the world and said, hey, I did this and, it's already done. And what do you think? Hey, by the way, so it's not, that's not how scientific research with human subjects typically is done. It's there's a review process that has to be approved by an internal ethics board at most research facilities, and only once that is approved do experiments go forward. And he pretty much circumvented all of that. And the other thing is, it now appears that he's missing the researcher. He apparently was doing this without the approval of his institution in China, and those are state-run institutions, and so I'm sure he was in violation of some sort of law and has now disappeared. So appears China is taking this quite seriously. Hey, that's it for this video. As it goes without saying, please like and subscribe. Let me know in the comments if you'd like to see more videos in this sort of more free-form vlog format. I'll provide links to all of these sources in the description. Thanks for watching, and I'll see you next time.